All right, time to talk about cell surface proteins. What makes these immune cells unique? Remember when we talked about clusters of differentiation? Well, that's what we're going to talk about now. So I'm going to show an immune cell, and you're going to tell me what unique receptors are on its surface. So let's start with T cells. What do you think's on its surface? Well, we know what the T cell receptor is, right? We know that if it's a CD4 helper T cell, then CD4 is, or if it's a cytotoxic T cell, then it's CD8. Okay, very good. Also, a CD, a cluster of differentiation that is unique to T cells, is CD3. Okay, and there are some other CDs as well, like remember CD28 that bound with what on macrophages? B7, very good. Okay, and CD40 ligand that bound with CD40 on B cells. That was involved in, what was that involved with? Class switching. Very good. So that's T cells. Now B cells. So we just mentioned that CD40 was on B cells. We also have immunoglobulins. MDs are mature, so IgM and IgD. They also have CD19, 20, and 21. That's unique to B cells. Okay, and then they have B7 as well that can complex with the CD28 on T cells. Macrophages, we just said B7, right? They also have, there's a receptor that actually binds to endotoxin on bacteria, and that is CD14. Very good. That's like a toll-like receptor. Okay. They also have MHC2 complexes as well. And finally, natural killer cells. What are unique about natural killer cells? 56, CD56, know that one. They also have CD16, and they have MHC1 receptors, as do all nucleated cells. So, time for a flash quiz. How do we differentiate immune cells? We just said that, by cell surface proteins. All right, now to introduce a concept called Anergy. Anergy means without ergy, or without ergo or zest for life. And that's what happens to immune cells sometimes. Why would that happen? Well, sometimes they can lose their stimulation and become non-reactive under certain circumstances. When would you want them to be non-reactive? How about if they are attacking your own cells, right? Right. That's when your body says, no, you should not be able to react with my own cells, so stop it. I'm going to make you useless. That's what Anergy is. Okay? Pretty straightforward. All right, so this slide is going to be a micro review. So do you recall, back to microbiology, what toxins and what bacteria cause massive cytokine release by binding to the MHC receptor of macrophages and T cells, the T cell receptor, and complexing these two cells together, forcing them to interact and co-stimulate one another. So here's a toxin doing that. And thus, they release a massive cytokine storm and leads to systemic inflammation all over the body. What is that syndrome called? Toxic shock syndrome, right? Or toxic shock-like syndrome, if it is. What bacteria cause those? Staph aureus and strep pyogenes. Very good. So definitely remember that from your microsections. What type of cytokines are we talking about here? Which ones cause the massive inflammation? We're talking like interleukin 1, interleukin 6, and TNF alpha. Okay? Also know the bacterial toxin endotoxin, which we talked about before. Again, and we just mentioned that this can bind to what receptor on macrophages? CD14, right? But this can bind to and stimulate macrophages to release cytokines, but also it causes what? What does it activate? What pathway? The complement pathway, right? And what specific type of complement? The alternative complement cascade. Very good. So now a brief word about antigenic variation. Now obviously, if you're a pathogen, the last thing you want to do is be detected by the body's immune system and killed off, right? So what do some of them actually do? Well, they actually change up some of the proteins on their surface. So for example, if it looked like this, they change it to look like this, so that immune cells become confused and don't recognize them anymore. That is through antigenic variation. And how exactly do they do this? Well, they can actually change their DNA. Remember with VDJ recombination, immune cells changing up their DNA to make different antibodies? Well, pathogens can do the same thing. They can splice out DNA segments and attach random segments together. They can also, so after it's transcribed, they can also splice out and rearrange their RNA segments too to make unique cell surface proteins. And then, what's the purpose of that? Then they can be undetected by your body's immune system. Kind of sneaky, right? So what organisms do this? Well, I'm not going to go through all of them, but just think of Salmonella with its flagella, Neisseria with its pili, Borrelia, and even some viruses like the flu with H1N1, and trypanosomes. That's a protozoa, so that can do it too. All right, the concept of passive versus active immunity. So we talked about this earlier when we mentioned that when infants, they're not able to make their own antibodies yet, so mothers actually help them by giving them antibodies. They even gave them to them in the womb through the placenta. They also give them after birth through what? Through colostrum, through breast milk. And they're given what type of antibody specifically? IgA, right? Right. Okay, so essentially the main difference between passive and active is, are your immune cells being passive or are they being active? So if they're active, then your own immune cells are actually being stimulated and making antibodies themselves. And this leads to a more robust immune response and a greater memory to pathogens. It makes sense because if you think about it, when the pathogen is processed by your own cells, like we see here, presented on the surface, and your own T cells are stimulated, they'll release cytokines that will lead to memory formation and a more robust response the next time you're exposed. This won't happen if you're just passively given antibodies. You won't know what the heck you're reacting against because they're not your antibodies to begin with, right? So we just said that when you have active immunity taking place, the next time you're exposed to that pathogen, you have a robust response. So wouldn't it make sense then to mimic a pathogen so that when you are exposed to the real pathogen, you have a strong response to it, right? Well, that's what vaccines are, okay? And there are two main types. There are live attenuated vaccines that are like the real live form of pathogen, but they're just weakened. And those involve, that's the closest thing to the real pathogen as you can get. So you consume, your immune cells or your normal cells consume the pathogen or the pathogen-like substance, and it gets processed the same way as a pathogen would, presented on the surface just as we saw before, and your immune cells, your T cells, are stimulated to the max, fully stimulated, release cytokines that induce the best memory. Okay? Another type of vaccination is inactivated vaccines. So these are actually not living at all. And this is close but not very similar to the real thing. This is just when we take a little part of a pathogen, heat up a bacterium or something, and take a fragment from that bacterium and expose your cells to this. So it's not the full thing because it's not degraded properly like we saw before, and so we don't have quite as good of a response. But this will stimulate B cells still. It'll still stick on the surface of B cells to cause them to make antibodies, right? So you'll have a lot of antibodies around, but you won't have the strong memory response that you would have had if T cells were involved. You need T cells for memory. All right, it's about that time again for a flash quiz. So, what are examples of killed vaccines? Well, you can remember them with the mnemonic RIP always, because they will stay dead. So they are rabies, influenza, and polio, as well as hepatitis A.